All right. Thanks everyone for being here. So I'm really excited to introduce Lindsay Campbell. She is an assistant professor in entomology and nematology at the University of Florida, based at the Florida Medical Entomology Lab. She's an interdisciplinary scientist trained in geography, ecology, and evolutionary biology, as well as public health. Her research sits at the intersection of data science and the environment, where she seeks to understand and predict the ecology of vector-borne disease systems. Her lab integrates and synthesizes data from multiple sources across sectors and develops spatiotemporal models to address fundamental questions about the distributions, abundances, and phenology of ecological systems across scales. This work bridges ecological theory with applied outcomes, and the ultimate goal is improving human and environmental health. All right, with that, I'll turn it over to Lindsay, and we look forward to hearing what she has to say.
Okay, I'll go ahead and share my screen. Thank you for the introduction and for the opportunity to present some of the work that we've been doing in my lab. Let's see. Okay, so I'll go ahead and get started. And so today I'm going to be talking about kind of broad scale assessment of abundances and phenology and how you can use neon data for investigating these types of questions. And so before I even get started, I want to make sure to highlight that the work that I'm going to present is really collaborative across not just students in my lab, but also other colleagues here at University of Florida and University of Kansas. And I also wanted to acknowledge all of the NEON staff and all of the people out in the field collecting these mosquitoes, identifying them, and working to record them and provide them for data analysis. So this talk, I'm going to give a little bit of background about the NEON mosquito program, just so people are oriented to the collections that we were using in analyses. And then I'm going to highlight an analysis that we performed investigating spatial synchrony of a West Nile virus mosquito vector across kind of broad scales. And then that sort of led into developing or deriving a nighttime female mosquito data set from the full mosquito collections. And then we actually haven't quite gotten full into phenology yet, so I'll just highlight some future directions on that component. So just a little bit of background. So the mosquito collections through NEON have started collecting mosquitoes in 2014, and it's been going on until present. And these mosquitoes are collected at 20 terrestrial core sites and 27 terrestrial gradient sites. They're collected using CO2 baited light traps with no light. CO2 is a really good sort of general lure for mosquitoes, and it can attract a large diversity of mosquitoes. Not every single mosquito on the planet, but a good diversity. And then there's also day and nighttime collections of these mosquitoes. So typically there's 10 plots at each NEON site, and so there's 10 mosquito traps across each site. And the sampling of these mosquito collections follows a field season and then an off season. So during the field season, the terrestrial core sites are sampled every 2 weeks, and then the terrestrial gradient sites are sampled every 4 weeks. And this goes on until there's 3 weeks where there's zero counts in mosquito traps, so no mosquitoes are collected. And when this occurs, which it doesn't occur across all of the sites, but if it occurs in more northern sites in particular, then the sampling scheme switches to this off-season sampling scheme. And here, instead of 10 traps being sampled every single week, at the core sites, just 3 traps are sampled, but they're sampled weekly so that they can get the precise timing of when mosquito activity begins again. And once it begins again, then the field season starts again. So when the mosquitoes are collected in the trap, then they go through a series of steps for taxonomic identification. And then a subset of the mosquitoes are identified and counted, and then you can estimate abundances from that subset of mosquitoes. And then there's also information about the amount of time that the trap was set out, which is pretty standard across sites, but the exact timing is available so you can adjust these estimates for trapping effort. So for us in my lab, we're super excited about having the opportunity to work with these data. It provides continuous monitoring across all these different ecosystems and domains. It's a standardized protocol. Also, the neon trap locations are typically distributed across more natural environments than what we typically see with mosquito monitoring programs. And, that makes sense because mosquito monitoring programs are trying to understand which mosquitoes are close to people and are there high abundances within vicinities of people. This provides a different vantage point to really understand the ecology and dynamics of these populations. And so these environments are undersampled. And then, of course, everything is open source through the NEON portal. And so in my lab, we're focus a lot on zoonotic vector-borne disease systems. So these are arbovirus systems like West Nile virus or others where there's a host, vector, and pathogen. The host can be hosts other than humans. And these are really complex systems because really the host, pathogen, and mosquito have to come together in space and time in order for transmission to take place. They're also complex because each of the components in these systems can have their own population dynamics. And each of these components are impacted by these external environmental conditions. And so things like landscapes, climate can alter the timing and distribution of when these components are coming together. And all of this affects transmission across different areas. So what we try to do is identify some environmental correlations and study the distributions, timing, or the interactions of either individual components or how these come together in order to understand precursors or to be able to predict a little bit better where there could be elevated hazard for some of this virus activity. So one virus that is a really important virus, really globally, but also here in the continental US is West Nile virus. It's a zoonotic arbovirus system. It has multiple avian hosts and also multiple mosquito vectors. It's the number one cause of arbovirus disease in the US. Once it arrived in 1999, it moved quickly across North America. And so I kind of mentioned that there's multiple mosquito vectors in this system. And this makes it particularly complex because each of these mosquito vectors can have its own habitat, its own phenology, its own population dynamics. They're generally distributed kind of regionally into these four sectors and sort of there's quite a few mosquito vectors, but they are dominated by these Culex species that occupy these different habitats. So despite all of these heterogeneities in these populations, habitats of mosquitoes, climate conditions, timing of everything, different seasons, occasionally there are really broad scale outbreaks in humans or spillover transmission to horses and other animals that you can see across these really broad areas. And so this is an example of these detected cases in humans and I think horses as well, accessed through the CDC ArboNet. And it just shows that there can be a lot of variation across years and that some years it's pretty widespread. So this kind of led me to think about this question of these broad scale processes that could potentially be drivers of virus transmission or activity. And so one way to approach this is to think about spatial synchrony. So spatial synchrony is an ecological phenomenon where there's similarities in temporal population dynamics across locations. And so this has been shown in multiple systems where sometimes across really broad geographic areas, there can be synchrony in these population dynamics. So the map on the top would show asynchrony, where maybe in some locations there's lower population values or abundances, and the map on the bottom would show where there could be synchronous population dynamics across timescales in these different areas. And so this has been shown in spongy moth populations or large bud moths, kind of these different pest species, but it had been less studied in vector-borne disease systems. So one of the challenges is that usually the analyses that you perform to be able to quantify this really requires consistent time series data over a relatively longer time period. And so this is where the NEON data really came into play for us, because we realized that there could be a continuous time series of mosquito vectors at these different locations, and we might be able to use those to go ahead and examine the potential for spatial synchrony in mosquitoes. So the first objective was just to see whether or not there was spatial synchrony present in the population dynamics of Culex tarsalis mosquito abundances. So Culex tarsalis, in kind of that 4 square region area map before, is a little bit more kind of a main vector in the western US. However, it has a pretty broad distribution, so we decided to go ahead and go with this species just as this first kind of look. And then the second objective was to see whether or not, if there was spatial synchrony in Culex tarsalis mosquitoes, whether or not there were any environmental variables that could predict that spatial synchrony. And so this synchrony can occur over multiple time scales. So it could be seasonal, which you might expect, although it could be over longer time scales. And so this is a nice way to try to examine the population dynamics across not just local spatial scales, but across multiple time scales. So in order to do this, we went ahead and worked with the raw neon mosquito data. We calculated the mean number of Culex tarsalis per month, and we ended up using data from 2016 to December 2019 because it was the most consistent across these locations. And the program was kind of beginning in 2014 and then started picking up. So this provided a good amount of time to be taking a look. And so the first thing we did was we identified sites with at least one Culex tarsalis record, which was 26 sites across. So these are neon sites. We went at the site level, and then if any of the plots with the traps, the 10 traps within them, had a Culex tarsalis, we counted that at the site level. And then we checked for this temporal coverage, and we ended up with these 9 locations. And so these locations are the monthly mean number of mosquitoes adjusted for trapping effort. And here you can see the distribution of these monthly mean number of mosquitoes per trap hour across each of the locations. Next, we accessed climate data, so daily climate data from GridMet, and these are at a 4-kilometer spatial resolution. And so we downloaded minimum and maximum temperature and also precipitation, and then vapor pressure index values, which are a measure of humidity. And then we aggregated these to monthly means and cumulative precipitation to match that same monthly unit that we had with the mosquitoes. Then I was working with my colleague Dan Bruman, who's at University of Kansas. He's an expert in spatial synchrony analysis and has developed multiple tools to be able to analyze spatial synchrony in different ecological systems, including this WSYN package in R. So I wanted to share this and the GitHub to this. And I'm not going to go really strongly into the exact methods that were used, but this package has a lot of resources and it provides all of the information about math and also has a nice vignette so you can go through examples. So in order to do this, we were using something called wavelet phasor mean fields. And so you have to, when you're working with the time series data, we needed to first demean the time series and then also detrend the time series and then transform the variance so that it was standardized to 1. So that was done across the Mosquito, Culex tarsalis time series, as well as each of the 4 or 5 environmental variable time series. Then again, we are using these wavelet phasor mean fields, which essentially is a way to decompose these time series across space and time into these in a way that you can analyze the periodicity across the full time scale, or sorry, study period and time scale. And so this, I'll go ahead and kind of orient everyone to this. So essentially what this is showing on the bottom is the full monthly time series of the data. So the 2016 to 2019 time period across all 9 of the sites. And then the time scale on the y-axis is the decomposition of the synchrony across these different time scales. And so up at the top, you can kind of see at the 12-month time scale, this would be an annual time scale. If it's red, that means that there would be strong synchrony. So the populations are fluctuating in a similar way at a yearly or annual cycle across the full time series that you see on the bottom. So this is really kind of thinking about how are these fluctuating together across space and at these different periodic time scales. Okay, so or looking at the Culex tarsalis, we could see that there was this, it's not surprising, it's mosquitoes. They might have an annual time scale that all of these locations might have similar fluctuations at an annual periodic time scale. And so really here it was more over this 10 to 14 month time scale. And then we also saw this area that was at about the 30-month mark or so, where there was just this little blip of synchrony down at this about 3-month time scale. And so this was just an isolated event. When looking at the temperature, we could see for minimum and maximum temperature, again, across the kind of annual time scale and even a little bit more extended, at 8 months or so, that there's strong synchrony in temperature across these different locations at that periodic time scale, which again, temperature is quite sort of annual and seasonal. It's not that surprising to see. For humidity, it was a little bit less pronounced across the full time series of the data. However, we did still see some areas where there was synchrony across. And then for cumulative precipitation, there wasn't really strong synchrony across all of these locations at the same time scales. However, there was this little blip down in this sort of isolated 2 to 3 month time scale zone, and it was sort of similar time period as that other small blip that we saw with the Culex tarsalis mosquitoes. So the next thing that we wanted to do was to see whether or not the synchrony in the environmental variables could predict the synchrony in the Culex tarsalis. And so here we found that minimum temperature was a significant predictor of spatial synchrony in Culex tarsalis. And so again, that's not entirely surprising when you think about mosquitoes and overall minimum temperature. This is, mosquitoes and other insects are ectotherms, so they can really be responsive to outside temperatures for their activity, development, if they're overwintering. So it's good to see this, but it was not a surprise. And then again, this is just kind of showing that yes, there is spatial synchrony in the minimum temperature values at the same time scale as the Culex tarsalis mosquitoes. So then kind of going back to this precipitation, we did not find significant, spatial synchrony in the precipitation, but really the way that these analyses work, it's looking to see whether or not there's significant synchrony at a time scale, but also whether or not it occurs across a broad range of the actual time series that you're working with. So I decided to go ahead and just take a look and see what might be going on, if there's anything that looks unusual or different about this specific time period, essentially, with the cumulative precipitation and the Culex tarsalis mosquitoes. And so what I was able to see going back and taking a look at some NOAA maps was that there was extremely high precipitation during this same time period across many of the locations that were sampled for Culex tarsalis. And so this is just kind of a correlation and an eyeball look, but we did see that, perhaps it's possible to pick up some of these sort of short bursts when there is something like an extreme precipitation event, and that you can see this across very broad geographic scales. So in summary, this was really kind of a first analysis of trying to look at spatial synchrony in a West Nile virus mosquito vector. It wasn't a huge surprise. We found that there was spatial synchrony in the minimum temperature values, and that those were predictive of synchrony in Culex tarsalis. The punctuated synchrony did coincide with that extreme weather event situation. However, there needs to be more analysis to be able to determine whether or not that might be whether or not there's an actual real correlation between those. And then overall, we're just kind of trying to think of things in the context of how it's possible to start examining mosquito population dynamics, not just at local scales, but how are these distributed and varying across these broad geographic regions. And so particularly for these types of viruses like West Nile virus and some other systems that do occasionally show these really broad scale or macro scale patterns, starting to look at these across both different spatial scales and also different time scales could be potentially useful for starting to disentangle some of this a little bit. So we do have a manuscript that outlines all of this work, and there's also a markdown file for everything that we did. And so Amy Bauer was really essential in putting the markdown file together, and that can be accessed through the lab GitHub. And it has the data as well as all of the steps that you can take to reproduce this analysis. And you could also use this markdown file if you wanted to try something on your own. And then again, Dan Ruman's lab has developed all of these tools. And so the WSYN package is really nice and has quite a few great vignettes to be able to examine or try these different analyses further. So that kind of leads me to this next part of the talk, which is going to be kind of just a quick overview. As we were going through everything working with the Culex tarsalis mosquitoes and kind of deriving that time series data set from the raw neon data, we realized that some of the steps that we were using might be useful to go ahead and derive a nighttime female mosquito data set from the full neon data. And so the reason that this is important is so that we could go ahead and adjust estimated abundances for sampling effort, but also to include zeros when sampling events took place, but perhaps no mosquitoes were collected, so it wasn't recorded. And also there's quite a few additional kind of nuances and notes that are in the NEON data. And so we ended up working with Sarah Paul and other colleagues at NEON to drive this data set and come up with a little bit of a best practices approach. And so I kind of mentioned first that there are these additional insights in these notes. So the raw data, usually does not include zero counts if there were no mosquitoes collected. And so the main step that is required is to go ahead and be able to infer a zero for a species or for an entire, site if a trap was placed but no mosquitoes were collected. However, sometimes there could be some notes in this data set, for example, because we're also testing for pathogens and there are things like a cold chain and other factors that could sometimes flag collection as compromised for if the cold chain was broken. But for us, we were interested in the abundances, and so we weren't that concerned about something like a cold chain being broken. So what we were able to do was dive into kind of some of these notes and differentiate some things between this cold chain, but also take a look and see what might be going on with individual trapping events that could potentially be important when considering what you might be interested for estimating abundances. And so we sort of came up with this, we didn't sort of, we came up with a set of exclusion criteria for trapping events where we would go ahead and remove a trapping event. For example, if the trap was tipped over, sometimes there's things like ants in the traps that are feeding on the mosquitoes. Sometimes mosquitoes they were kind of frozen to the edge of the trap, and so it wasn't possible to get a full estimate of abundances of different species. And sometimes when traps are placed out, there's things like interference with the carbon dioxide flow out of the trap. And so if there wasn't kind of an equal lure amount coming out of each trap, then we considered that to be compromised. And then in some cases, people had notes like they went to the field and there was an angry cow, so they couldn't pick up their trap for that trapping event. And so again, I touched just a little bit on inferring these zeros. And, we really wanted to be able to have these zeros in these time series because I think this is one of the great advantages of this continuous trapping effort is to really be able to have continuous time series, but then also being able to identify the beginning of adult mosquito flight activity. And it also opens up the door to be able to use multiple different modeling approaches when trying to understand these distributions. And so again, I'm not going to go into all the details of what we did here, but we spent quite a bit of time working across these data. We ended up taking the notes from the sort of free text notes from different like trapping events and taking a look at those and clustering them into different categories and open refine. And then we developed a derived data set that is a data package available through the Environmental Data Initiative. Again, Amy Bauer was really essential in developing this, and it has a markdown file so you can see all of the steps that we took in order to be able to drive this data set and what our exact exclusion criteria was, and it's reproducible. So just as a quick glance then across these sites. From this derived data set, we can see following some rarefaction for species richness. This is the species richness that is occurring across all of these different neon sites. There are about 5 sites that were excluded following rarefaction. You can also see this basic diversity index for Shannon's diversity index over kind of in the eastern portion of the state or of the US. There's greater Shannon's diversity across these different areas. And then also just taking a look at Simpson's diversity. And so, really, when we take a look across the number of different mosquitoes or sites that these different mosquitoes are distributed across, we can see on the left that there's multiple species that have pretty broad geographic distributions across multiple sites. And then, as expected, there might be some that are very locally distributed. But I just wanted to note, even looking at these sort of top 20 or so species, there's multiple medically important mosquito vectors or species in this list. And so this is quite interesting and fascinating. It shows that these neon sites in these more natural areas are collecting these medically important mosquito species. And for us, it kind of reinforces that more natural area and provides a great asset for being able to understand the ecology and the dynamics of these species better. So I mentioned at the beginning that we haven't gone fully into phenology yet. However, we have fit phenological curves for all of these species at the neon sites. But right now, it's essentially just a bunch of pheno curves, so I didn't want to present that today. But we'll be performing some analyses, and we're particularly excited about looking at differences in phenology over time, both within species, but across locations, and also with different mosquito traits. And one thing that we are working on is some tools to really start to try to pull out phenological or phenometrics for mosquito species and other organisms that have more than one generation per year. And so a lot of the phenometrics that exist currently kind of assume that there's in one single generation for the year. For us, we're particularly interested in the second or even third generation population curves, and if there's any environmental drivers that might be driving those or precursors to some of those later season population dynamics. For us, it's quite interesting in the context of thinking about transmission, but overall, I think it's just fascinating thinking about this in the context of ecology. And so this is coming to the end of the talk. Here are some references for different items that I talked about. And then again, along the way, there's multiple papers and references that I included in the presentation. Okay.
Right. Thank you so much, Lindsay. That was really, really interesting stuff. I'm excited to see that in its presentation format. Yeah, so if anybody has questions, please feel free to add them to the Q&A. If it's, you can add them to the Q&A, or if you'd like to raise your hand, we can have someone on the Neon side unmute you and let you talk, especially if it's a more convoluted or longer version of a question. But I am not seeing anything yet, so I can kind of get things kicked off. I mean, I have lots of questions. One of the ones that kind of popped to mind as I was thinking, I really like that synchrony paper, and I think I sometimes have a hard time envisioning what the models are actually telling us. But I was kind of curious, I think of West Nile virus as having good years, or a good year being where we don't see it as much, and then a bad year. And I know that people have been attempting to explain that for across
Great.
decades in papers and all that. And I'm wondering if you ever thought about like thinking about spatial synchrony, not just of the vectors, but like also of the hosts, and if that might help kind of explain bad years or certain regions where you might see something worse. And if, and it might not even necessarily be mechanistic, right? It's not like the birds are responding to some environmental driver. It just happens to be that they're both, becoming more or less abundant at the same time. Anyway, I didn't know if you'd ever
Sure,
thought about it.
yeah.
I've seen papers on that topic.
No, I'm glad that you answered or asked that question. And so spatial synchrony is kind of, it can be really abstract to try to, because you're trying to think about, and it actually is a little abstract to try to describe, but you're trying to think about how might these populations be fluctuating in similar ways across these different environments. And so in the context of thinking about that, there's a few different things that can produce those patterns. And so one is intrinsic population dynamics. Dispersal is another one, or these environmental variables, which are referred to as Morana effects. And so in the context of West Nile virus, there's sort of this local synchrony between avian hosts. So perhaps the timing of avian hosts and overlap with mosquito abundances that is really important for transmission. And so this is an extremely complex system. So so thinking about it in the context of what might be producing those broad scale patterns, it could be a combination of the synchrony in the bird populations and when there's a lot of susceptible hosts, maybe at a similar time scale across these really broad regions. But it could also be from dispersal. So if there's both migratory and resident species that are kind of these hosts that are considered important for this virus. And so it's sort of like a lot of moving parts all together that could lead to or produce this broader scale kind of activity than you might see other time periods. So I don't know if that answers your question satisfactory. We were trying to just look first to see whether, what's the situation with the mosquitoes? Can we take a first pass at this? And we had considered trying to look at the avian hosts as well.
That's really cool. Yeah, I've always had just sort of an inherent interest in the timing of those interactions. And I think especially in West Nile where the nestlings can be really important amplifiers and things that it seems like it might be a cool next direction. I just wasn't sure if what I was saying made sense, but it sounds like it does.
Oh,
So,
it does make sense.
okay.
And yeah, we're also excited about the avian hosts. And one other thing I'll note is that particularly one of the things that we're excited about is the opportunity to sort of integrate data across different data streams and sectors. And so thinking about here is, neon data for long-term, ecological monitoring. But there's also, growing sets of community science data such as eBird and some other efforts that can also start to be used to think about this across space and time a little bit more. And each one is obviously going to have challenges and how you integrate that and, some biases, but I think there's a lot of opportunities on that front to start to get at some of these mosquito vectors and also hosts together.
Excellent. Well, great. So we've got some great questions trickling in as well. One of them is awesome work. I'm curious whether there are any known differences between mosquito species in factors that would influence capture rates, like dispersal distances or attractiveness to the CO2 traps.
Sure. So I'm not an expert in every single mosquito species, but so here, there's a standard trap. So different Culex species are attracted a little bit differently in particular to these CO2 baited light traps. A lot of times they are collected in higher abundances in something called a gravid trap. However, what we were looking at here is we weren't really comparing across species for the abundances. And because it's a standardized trap method, it's sort of relative to this trap. And so in that context, we felt like the differences or fluctuations are likely they're similar, across these different locations. But there are different flight distances for different mosquitoes, and there are also different attractants and trapping methods that are a lot more efficient at capturing specific species, including multiple medically important mosquito species.
Excellent. Thanks so much. The next question is from Will Hendricks at Neon. It says, thanks, Lindsay. Did you consider using Neon Instrument Systems data for the meteorological data? And if so, what made you choose the GridMet data instead?
Yeah, so actually we chatted about that sort of after the fact. So we, in our group that we were working with, we all had quite a bit of experience with some of these gridded climate data products. And so GridMet or DayMet and some others. And so that was kind of, we went, I think, with that because that's what we knew really well. However, since that time, we've learned a lot more about the different environmental data sets and sensors, obviously, at each neon location and site. And so we're definitely interested in integrating those types of monitoring efforts on the environmental side with these biological collections.
Okay, great. Thanks so much. Okay, so the next one is from Kate Tebow, also at Neon. Thank you for the great talk. And have you looked at synchrony across species to explore how interspecific interactions might impact the dynamics? So it's related to the question I had asked, but more
Mhm.
focused on different mosquito species.
Yeah, so we're actually really, there's so many different questions that you can look at with synchrony. And so you can look kind of at community level synchrony. There's been work done, I think, by Jonathan Walters looking at species, synchrony and species richness. There's multiple pathways. And for us, we found that it was probably would be beneficial to drive this full nighttime female mosquito data set first, and then that would open the door to being able to start to look at some of these questions across multiple species or even, looking at things kind of in different regional dynamics.
Yeah, I was thinking the same thing while I was thinking we have so many species to look at. It'd be really cool to to
Yeah.
get all those. Okay, next up is from someone in Domain 3, which is down in the Florida area. Do you know if the phenology data is affected by ENSO, the El Nino Southern Oscillation? I know
Oh,
some of
yeah.
what your analyses focused on were more on the western side, but anyway, wait.
No, that's a really great question. And I'm glad that you brought that up because I think it can put some of this timescale decomposition into a little bit better context. And so sometimes there's these sort of oscillations or periodicity in any time series, or in the synchronous time series that are related to things like teleconnections. And so is, considered a teleconnection where you can have sometimes a response, for example, in weather or climate over a season in a specific area where you can see responses from that. And those are typically, I've been worked with people on papers who are looking at, you can't predict exactly when INSO is going to happen, but those are a little bit longer term periodicity or oscillations. And the reason those types of lags are really interesting and useful to think about is because, if you can start to kind of identify those out in front a little bit, it gives you some lead time to begin to kind of anticipate or think about what might be happening in the near term future. And so anytime you can find that there's some lag periodicity in a system, then you can use that to start to think about how can I disentangle this? Is it just intrinsic population dynamics, or is there an association with a climate variable? And can I use that to make a prediction? So looking at that across multiple time scales and spatial scales is kind of exciting because we see these processes are happening across all of these scales.
Cool. That's a great answer. Okay, next one is from Tanya Maslak. Thanks for the interesting talk. Could you speak more to the potential for these findings to be translated into applications for public health? Is there potential for early warning systems or more targeted resources for integrated vector management efforts at the local level or other potential benefits?
Sure. So this particular analysis was kind of a first pass. And this one, I'm going to say it was more on the ecological knowledge side. And so, however, I think all of these types of analyses contribute to the broader knowledge of system dynamics and what's taking place across different scales. However, I think that the neon mosquito data, again, is particularly for us exciting. We also work with a lot of mosquito control program data that people have worked really, really hard to collect over long periods of time. And some of those data sets are now available through some open source, well, like through VectorByte and some other, which is open source platforms. And we can integrate this neon data with the vector control program data. And so essentially what that means is that it gives a broader view of these species across these different environments, which helps to improve models and also then provides a little bit better context for making predictions or understanding these population dynamics. And so, well, this paper does, I'm going to just say no, it does not translate directly to vector management. I think that all of these approaches are useful for trying to get improved management.
Great. That's a good question and a good answer. So next up, again, from the D3 folks, they were asking about whether you ever see hurricanes impacting the data. And I think maybe the specific sites you were looking at for Helix Darsalis, maybe not, but I didn't know if you'd ever
Yeah.
looked at some other.
So with the neon data, I have not looked specifically at hurricanes. There were quite a few hurricanes just a couple years ago, and so it'd be possible to look with the neon data. For the mosquito control program data, you can look at that data. Often the for people who have decided to share that data, there sometimes understandably, there's disruptions to monitoring and abatement and control activities, or they have to move into kind of a different mode to deal with some immediate situations. But anytime I think that we are starting to work with this longer term monitoring and time series, then we can start to understand better how some of these perturbations from these environmental events impact even a little bit later downstream dynamics of systems.
All right, great. Yeah, I remember, I think it might be one of our taxonomists had some photos after a hurricane of just buckets and buckets of those floodwater mosquitoes. So yeah, great. Well, I think, hang on, there's one more. Okay. One more Q&A. So this one's from Courtney Meyer. It was interesting to see the potential impact of precipitation events on your results. Did you also investigate the lack of precipitation? So drought events.
Sure. Yeah, and so that's a really good question. So when I first saw sort of that little blip, all it was saying was that there was kind of this punctuated spatial synchrony in the abundance values across those 9 locations at that 2 to 3 month time scale at that specific time period. So I didn't actually know. I didn't know if it was anything, and I didn't know if it was which direction that might be in. So it could have been in extremely dry, or it could have been, in this case, what looked like this punctuated precipitation, heavy precipitation event. So I think that both of those types of things warrant further investigation. And in talking with my colleague, there's a couple probably better routes to go on the modeling front to try to look at those types of events. But we did see just a little bit of this approach did pick up, this little event there.
All right. Well, that's fantastic. That's all I'm seeing for Q&A at the moment. And I think that's probably good timing because I think there's a few other notes that the organizers wanted to make. But thanks. Let's all give Lindsay a round of applause. That was a wonderful talk and great answers.
Yes. Thank you so much. Wonderful presentation. Very excellent use of neon data. We thank you so much for sharing your work with us today. Everyone, thank you so much for attending the seminar. I put the link in the chat, but we have a lot of more exciting events coming up for you. We have our neon research support webinar at the end of March, and then another science seminar on stream drawing coming up in April, as well as a data skills webinar where you can learn more about how to work with the data sets like Lindsay was telling us about today. So we'd love to see you at a future event. Thanks again for being here. And thanks again, Lindsay, for a great talk.
Thank you, everyone.